Hello to all students. I hope you are doing fine. This is Professor Masood fuzail. In this video lecture, we are going to discuss chemiosmosis. Chemiosmosis is a process of synthesis of ATP with help of movement of protons, which are usually known as H positive ions, across a semi-permeable membrane. Now let's discuss this process in detail. As you know that our cells utilize different mechanism to produce energy and this energy is stored in the form of ATP, and this production of ATP, the process of synthesis of ATP, is known as phosphorylation. There are different methods of phosphorylation like photophosphorylation and which takes place in chloroplast during the process of photosynthesis, but at this stage we are going to discuss chemiosmosis, this process of phosphorylation in mitochondria, because chemiosmosis can take place in mitochondria as well as in chloroplast and some bacteria. So first of all, we are going to look at the structure of a mitochondrion. As you know that mitochondrion is an organelle of eukaryotic cell which is composed of double layer. So this is the outer layer of the mitochondrion, which I kept simple, and this is the inner layer of the mitochondrion. Inner layer mitochondrion I have shown as a lipid bilayer. There are two layers, upper lipid layer, lower lipid layer. There are two layers of the lipids. Now, cell utilizes glucose. Whenever we eat any kind of food, it is converted into glucose, which is supplied to the cells of the body. When glucose enters in our cells, the process of glycolysis takes place and glucose molecule is broken down into pyruvate molecules. Then pyruvate molecules are converted into acetyl coenzyme A, and then acetyl coenzyme is entered in mitochondria where Krebs cycle runs. And after Krebs cycle, there is another process known as electron transport chain. All these processes are used to synthesize ATP present in the food molecules. Glycolysis takes place in the cytoplasm while Krebs cycle and electron transport chain takes place in mitochondria. Krebs cycle usually runs in the matrix, which is the central part of the mitochondrion, while electron transport chain runs on the membrane of the inner membrane of the mitochondrion. There is a space between two membranes, outer membrane and inner membrane. This space is known as intermembrane space. As you can see, this is the central part of the mitochondrion, which contain cristae and matrix, which is a kind of liquid, and there are two types of electron transport chain running on the membrane. So we are not going to discuss in detail Krebs cycle, electron transport chain or glycolysis, because today we are going to only emphasize on chemiosmosis, how this process takes place. Anyhow, whatever the process, glycolysis, Krebs cycle and electron transport chain, all these processes produce ATP. Total 36 molecules of ATP are generated during this process from 1 mole of glucose molecules. During this process, there are two other molecules which are also generated. One is known as NADH, nicotinamide adenine dinucleotide, and FADH2, which is flavin adenine dinucleotide. There are also high energy molecule which are used on the electron transport chain. So let's little bit review about these electron transport chain. Look at this diagram. This is a membrane, inner membrane of mitochondrion. Right over here, one molecule of NADH, which is a coenzyme, nicotinamide adenine dinucleotide, is going to be oxidized and it converted into NAD, and plus H positive ions are produced and electrons are generated, which are used to run electron transport chain, which takes place by different protein molecule present on the surface of the membrane, in which coenzyme Q, cytochrome complex, cytochrome C, cytochrome A, cytochrome B, etc. H positive ion which are generated from NADH are transported by these protein molecules into intermembrane space where concentration of H positive ion increases and pH becomes low. pH become low and concentration of H positive ion become high. Some of the H positive ion are also used to synthesize water by combining with oxygen during um, oxidation process during electron transport chain. Now let's look at this diagram. In this diagram, we are using FADH, flavanoadenine dinucleotide, which contain 2 hydrogen. It is going to oxidize into FAD plus 2 H positive ion and plus 2 electrons are released, which are used to generate electron transport chain. These H positive ions are also transported across the membrane from matrix into intermembrane space where concentration of H positive ion increases as a result of pH decreases. Some of the H positive ion are also used to combine with oxygen, free oxygen to form water molecules during this process. Now what are NADH and FAD? These are coenzyme molecules which are oxidized and reduced. During this oxidation and reduction, they produce electron, they accept electron and they release electron. Look at this diagram. This is an NAD molecule, nicotinamide adenine. It is going to reduce, it contain only one hydrogen atom when it accept one H positive ion plus 2 electrons and it is reduced into NADH molecule. Now at this stage it contains 2 hydrogen molecules. But this reaction is reversible and when NADH molecule oxidized, during oxidation it produces 2 electron, 1 H and NAD molecule. In the similar way, FADH molecule can also undergoes oxidation and reduction reaction. This is a molecule of FAD, flavanoadenine dinucleotide. It also accepts 2 H positive ion and 2 electrons and it is reduced into FADH2 molecule. This is also a reversible reaction and FADH2 molecule when breaks it converted into FAD, 2 H positive ion and 2 electrons. As you can see this part of the diagram. In this diagram there is no H present and in this diagram you can see 2H located on the surface of the FAD molecules. So these molecules are also generated during the process of breakdown of glucose during Krebs cycle, which are later used in electron transport chain to transfer electrons and H positive ions. So whatever the process, both of these electron transport generate H positive ions which are collected in the intermembrane space and as a result concentration of H positive ion increases in intermembrane space. Now this H positive ions which are protons, when accumulate they contain a kind of potential energy which is separated from inside the mitochondrion with help of a membrane lipid bilayer which does not contain any charge. So these ions are transported with help of an enzyme known as ATP synthase and used their energy which is in the form of potential energy which is converted into kinetic energy, and these ions, H positive ions, are move across the membrane to generate ATP molecule with the help of ADP and inorganic phosphate. ADP, adenosine phosphate, when combined with inorganic phosphate plus it is converted into ATP molecule by the energy generated by hydrogen ion. So because ATP is synthesized during the movement of H positive ions which are also known as protons across a membrane, that is why this process is known as chemiosmosis. Chemi means protons, chemicals, ions. Osmosis is the movement of, usually it is considered as water molecule, but right here we are only using this term for ions. Osmosis is the movement of molecules from higher concentration to lower concentration across a semi-permeable membrane. So right over here there is a semi-permeable membrane which is the inner membrane of mitochondrion which is going to be utilized over here, and movement of H positive ion is taking place from higher concentration to lower concentration, just like ions which get separated in a battery. As you know, there is a battery in the car which contain two types of ions, positive ion and negative ions. These ions get separated by a septum, by a wall. When these battery contain two terminal, positive terminal and negative terminal, when these two terminals are joined with help of a wire, and on the way you can put anything like a bulb, like a fan or anything you want to run. when ions move from because negative attract positive, so when ions move from opposite poles and attracted toward each other, the potential energy stored in these ions is converted into kinetic energy, which is then utilized, it is then used to generate different kinds of powers, like we can run a fan, we can run a motor, we can lit a light. So that is how this process takes place. So the energy stored in the H positive ion is utilized to produce ATP. So there are different methods of production of ATP. So as you know that in chloroplast, process of photosynthesis takes place, and during photosynthesis light reaction takes place, and during light reaction cyclic phosphorylation and non-cyclic phosphorylation takes place, which is known as photophosphorylation. And this photophosphorylation is coupled with chemiosmosis to generate more molecules of ATP, as well as this process of synthesis of ATP also takes place in mitochondrion to help to generate more ATP molecules, which are coupled with the other chemical reaction taking place in the cell. So I hope it makes sense, and I'll see you in the next lecture. Until then, bye.